So what I'm going to do is really try to introduce the Healthy Brain Network as well as some related efforts and just try to explain the motivation for naturalistic viewing for large-scale studies. So just about everything I'm going to talk about is already in publication or on bioRxiv, so that makes this easy. But here what I'm showing is the data descriptor and scientific data for the Healthy Brain Network, which is really an announcement paper that provides all information regarding study design, rationales, lessons learned. But in short, the Healthy Brain Network is a study that's aiming to collect data from 10,000 children and adolescents ages 5 to 21. It's using a community self-referred recruitment strategy, which basically comes down to these are generally kids whose family have some sort of concern, whether it be a behavioral problem, learning problem, some difficulty. And the goal really is to get a very large-scale sample that's highly enriched with respect to mental health and learning disorders. Of course, in the middle of all this, it's a transdiagnostic sample. So the idea is really to spend a broad range of samples. Why? Well, when we think of a lot of our challenges, it's not so much is a child atypical versus typical, but really it's how do we differentiate different presentations. Now that's something that with a large-scale transdiagnostic sample, we can really start to delve into. Also, the data sharing model on this is quarterly. So we've been sharing data for the last year, and that will continue throughout the course of the next 4 years as data acquisition wraps up. Up here, I'm showing from the first 1,000 subjects, just give you an idea of the range of diagnoses. I will say that right now we're over 2,000 subjects in, and I would say the profile will generally consistently match. Where, of course, if you're asking about, are you worried about your child? Not surprisingly, ADHD comes up quite frequently. But then you also start to see anxiety disorders, disruptive disorders, learning disorders, autism spectrum, and so forth. Here is the imaging protocol. This is a multi-site study. There are some small variations related to whether one is on a trio or on a prisma. But as you'll see, there's resting state, there's diffusion kurtosis imaging, but also notice that there are 2 movies, Despicable Me and The Present. These movies were selected to be emotionally evocative, and that's something that, given what we're after with trying to probe mental health and learning disorders, that was a key goal can we have something that might help to differentiate individuals? Okay, so really though, when we first look at what was our first motivation for movies, it was really about head motion. One of the challenges in going in any child population is head motion. Now step into a child population where half the sample has some form of ADHD, where you have kids with increased levels of anxiety and so forth. What we quickly noticed in some of the piloting is during resting state, we had a good number of kids who are like, I'm done with the study, I'm out. You don't get that so much when a child is watching a movie. And when we looked at the head motion estimates, we generally found that movies did have lower head motion when compared to rest. One thing I would also note is in the paper we make a point about mean FD versus median FD, noting that the mean FD often wound up being somewhat inflated due to extreme outliers. So that's just one consideration. One thing we try to draw attention to is as the sample size scales up, to really keep in mind, you're going to start pulling out phenotypic associations with your imaging data that may really be about imaging quality. So here we took Cameron Craddock's pre-processed connectome quality assurance protocol, and what we're showing is that for a range of psychiatric variables, there are a number of variables that you can wind up pulling associations with, which is just about scan quality, in particular emotion. You will notice that more associations seem to come out for rest than either of the 2 movies. Now, one of the other things that we were concerned about up front when discussing movies was this issue of how equal is the data? Can we still do functional connectivity analyses? So we carried out the Healthy Brain Network's serial scanning initiative, which was openly shared. This is one of these efforts to really get high characterizations of subjects, but we had 4 different states: rest, inscapes, movies, and flanker tasks. And one of the things that we noticed is the between-session test-retest reliability for edges seemed comparable with other studies, but when you get between condition, that's not where your greatest source of variation is. So for most edges, not all, you actually have quite high ICCs when you go between condition. It's the between-session variance that will remain a challenge. And of course, as you increase the amount of data that you feed in, then you watch your between-session reliabilities come up substantially. but that was reassuring, because one of the things is you want to know if this is something Tammy mentioned earlier, is the movie somehow making it where I might do worse? But actually, there are a number of publications now reporting they're seeing higher reliabilities with movies, and I think that's a highly relevant point. One thing I also wanted to draw attention to is we've been stepping into non-human primate imaging, and most recently, beyond just our own work with non-human primate imaging, we established a primate data exchange. This is myself, Daniel Margulies, and Charlie Schroeder. In Prime, there's over 155 monkey data sets that are now openly available, several of which include naturalistic viewing. In particular, there's Ting just published in Cell Reports a paper focused on parcellations, where we have awake and endosleep animals. And in the awake animals, we have both movies and rest conditions. Related to that point, we did an additional analysis where if you look at the distribution of frame-wise displacements for rest versus movies versus inscapes in the non-human primate, what you'll notice, you see notably more motion in the rest condition than the other conditions, which this has been something consistent that we found when we were stepping into awake imaging. The monkeys definitely tolerated better with movies. Now, initially we were using human movies, but more recently we've switched over to using monkey movies in order to increase the engagement, as one can imagine. One other point to make is that when we looked at the comparability of the parcellations that Ting's work wound up revealing, is when you looked at anesthetized versus awake, there are very large differences. But if you looked at awake during rest and awake during a movie, actually the gradients were more comparable. Once again, going back to the point that if you're doing standard functional connectivity analyses, they will not vary tremendously as a function of whether or not you have the movie signal mixed in to your data. One last thing I wanted to point to is beyond head motion, another concern that exists around movies is the person or the monkey actually watching the movie. Now, Steve LeConte many years ago developed something called peer eye estimation regression, or PEER, which Cameron Craddock, who really was a major force in putting the imaging protocol together, had included PEER. In short, what PEER is you have a subject look at each of 25 points for 4 seconds, one at a time, and you cycle through. And then what happens is you just take the signal from the orbit in your fMRI data, and you basically train a support vector regression model to go ahead and actually predict the location where someone is looking at each TR. And what's happening is because of inhomogeneities in the eye, this actually works quite well. What you're seeing on the right is, on the top is the ideal fixation locations, and then in the rows you have the fixation sequence for each of the subjects. And you'll notice a high consistency across subjects. You'll also notice that it gets fuzzy as you go towards the bottom. That's head motion. So as long as you have relatively low motion, you don't need an eye tracker. You could actually just use this 1.5-minute scan in your scan session and use that to actually look at where someone's looking. And finally, before Tammy kicks me off the stage, what I'm just going to show is that classifier applied to 2 different movies, The Present and Despicable Me, and you'll notice, once again, highly consistent responses across subjects. And it's so much so that you'll also notice there are differences between the 2 movies. Well, those are actually quite predictive. You can get an area under curve of about 0.9 for a prediction of what movie someone's watching based upon the PEER series calculated from the orbit. And finally, one other point is we use out-of-scanner eye tracker information if you look at the lower right, and you'll see that someone watching The Present in the scanner using PEER versus using an out-of-scanner eye tracker, highly comparable fixation sequence patterns. So this takes care of a major confound that will come up in studies of naturalistic viewing because you don't have an observable response. So here you do. So you can say the person was watching the movie, which will address questions on are individual differences being driven by whether or not someone's watching. And just many thanks to everyone involved, in particular with the imaging protocols, Cameron Craddock, and also Leai, who's in our audience, who's really been on the front lines of Healthy Brain Network as far as organizing the data and dealing with the many questions, and everyone else involved. Obviously, this is a massive team effort. Thank you.